Ph.D. in biophysics from Oregon State University, and after went to study as a postdoctoral fellow with Dr. Kenneth Pagan at the Roswell Park Memorial Institute in Buffalo, New York. He has joined the faculty at UCLA in 1979 and has been here since, lucky for us. And he has a joint appointment as a professor in the Department of Microbiology and Medicine, as well as he's the vice chairman of the Department of Human Genetics. He's an international authority on atherosclerosis and mouse genetics and has been a longstanding leader in the UCLA atherosclerosis research unit. So thank you very much, Dr. Lusis, for giving ground rounds today, and I'll turn it over to you.
Yeah, well, thank you very much, Christopher. I am not vice chair of human genetics right now, so
okay, apologies.
Yeah, so let me pull up the screen here. Oh, where was it? Can you see that?
Yes, yeah, we can.
Okay. So today I'm going to tell you about I guess a novel endocrine circuit between the liver and the heart, and I'll show you data that indicates that also plays a role in heart failure. Most of the studies I'll be telling you about are in mice, but at the very end I'll show you data suggesting that it also, the same circuit plays a role in heart failure in humans. And I want to mention that this is primarily the work of a very talented postdoctoral fellow in the lab, Yang Cao. And Yang right now is in Chicago at the American Heart Association meeting, presenting this very same piece of work. Also, I think it would be best if you ask questions along the way. So I think that if you have a question, why don't you just unmute and just ask as we're discussing this. Is that okay, Kristen? I don't know if Kristen's there.
Yes, that's fine.
That's fine. Okay. Yeah. So don't hesitate to ask. Okay. So our lab has studied common complex forms of cardiovascular disease, common forms of atherosclerosis, common forms of heart failure. And by complex, I mean that these disorders, the common disorders, are due to the interactions of many genes, as well as environmental factors, right? So obviously they're difficult to dissect. And our approach is sort of a population-based approach. We study natural variation that occurs among mice and humans, and we try to relate that to the various traits and also to molecular traits such as global gene expression, and then integrate the data to try to understand, susceptibility to disease, hopefully develop, diagnostic approaches or even therapies from that, and to really get an idea of the molecular pathways involved. So now, in terms of complex diseases, I think, as I'm sure you know, genome-wide associations studies have been really revolutionary. So, for example, this is a study, there's a more recent one actually for cardiovascular disease, but this is a study of about 185,000 people. It's actually a meta-analysis from multiple studies. And it shows the locations of DNA variation on the various chromosomes. And notice the X is missing here because that's hard to sort of integrate in a lot of these studies. You can do it as well. And on the y-axis is the negative log base 10 of the p-value. The significance, sort of a Bonferroni correction, is shown by this line. And there are about 160 loci that are causal for atherosclerosis. These loci usually contain multiple genes. In some cases, the causal gene has been identified, usually in studies in mice. In many cases not, but here what they've shown here are the most likely candidates at each locus, right? So this is great. This gives us a lot of different genes. It gives us kind of an unbiased view of atherosclerosis. There are genes here that affect lipid metabolism, cholesterol metabolism. There are genes that affect inflammation. There are genes that affect hypertension. And we can organize all these. And this really does tell us, here are the reasons why we develop. Some people develop cardiovascular disease and others do not. Each of these loci explains a very small fraction of disease. And certainly there will be many, many additional loci that contribute, that remain to be studied in larger populations, right? But, so this is great. But what we would like to do is to go beyond just identifying these causal genes and putting them into categories. We would like to sort of have we would like to know about the pathways they're involved with, how they interact with each other, how they interact with the environment, sort of a higher level of understanding of these genes and how they affect atherosclerosis. And for that, we have turned to mice, right? Studies of complex traits are simplified in mice. There are, of course, differences between mice and humans. And our strategy is to try to study mice, to understand the genes that regulate these traits, and then to look at human cohorts and ask whether the conclusions that we've made in mice are valid in humans. Right? Now, the way we study complex traits in mice is using a panel of inbred strains, which we call the hybrid mouse diversity panel. Right? We developed it about 15 years ago. It has the advantage over previous sort of analyses in mice in that it enables relatively high resolution association mapping. And I won't go into the technicalities of that, right? We look at about 100 strains, just so we have enough power to detect at least the strongest loci. Of course, we don't have as much power as a study with 200,000 individuals, but we can do different kinds of things. There are a number of advantages to studies in mice as compared to humans. In this sort of a design, this panel, we have replication. There are an unlimited number of mice of a given inbred strain. Right? I should have said that these mice differ. There are millions of differences, genetic differences across these strains. But we have replication. We have access to relevant tissues. In the case of heart failure, you can imagine that's particularly important. We can see the hearts, right? We can examine the hearts. We can do metabolomics and proteomics and so on. We can control the environment. We can sensitize the conditions. We can sort of, as I'll show you with the heart failure, we can create conditions that predispose to heart failure. And then the genetics can tip it this way or that way. So it gives us much, much more power than one would have in humans. And finally, we can do experimental validation. Okay. And again, if there are any questions at all, please interrupt me at any time. Okay, in addition to looking at the clinical traits, we also do what we call systems genetics. And again, it's using omics, which everybody uses these days, right? So we have genetic variation, we have the clinical traits that we're looking at, but in each of the mice, then we could also do global transcript levels, such as in the heart, or proteomics, or metabolomics, or even the gut microbiome. We can look at all of these things. We can then model. We can model pathways. We can follow genetic variation through all the way to clinical traits. We can look at correlations. We can model causality, causal inference. And we've done this with atherosclerosis for many, many years. Heart failure, we started looking at more recently with Jessica Wang when she was a graduate student, Christophe Rao, and so on. So today I'm going to tell you about only one project. This is this one about crosstalk between liver and heart. Okay, well, this sort of, we've been interested for several years in heart failure with preserved ejection fraction. And I'm going to relate the variation, the crosstalk between liver and heart to heart failure with preserved ejection fraction. And so I've listed just some characteristics that I've read about. I'm not a doctor, so I haven't seen patients with this syndrome. So I suspect many of you have a deeper understanding of this than I do. But it's very heterogeneous, that's clear. It's very common. Generally, the ejection fraction, that's sort of one of the definitions, is about normal. But relaxation, diastolic function is very often, or that's one of the criteria, is defective. It's often associated with obesity and other metabolic disorders. There's also very often atrial dysfunction. There's pulmonary hypertension. So there are a whole bunch of characteristics of this. One interesting characteristic which we've studied is sex bias. About 2/3 of the patients tend to be women. And this has been discussed. Why would this be? We think it has to do with mitochondrial function, but I won't talk about that today. Therapies for heart failure with reduced ejection fraction are not effective. There are some developing therapies for heart failure with preserved ejection fraction. But we have a long way to go, right, in terms of diagnostics, therapies, and so on. And the underlying mechanisms are largely unknown. So that's really our main goal, is what are the mechanisms that contribute to heart failure with preserved ejection fraction? One thing I should say here is that, in contrast to like lipid levels or hypertension or obesity or atherosclerosis, this genome-wide association studies have not been very successful with heart failure. This is a very recent study. You can see there are only a small number of loci. There are undoubtedly hundreds of genes that contribute, but the GWAS only gives us a small number, and undoubtedly that's because of the great heterogeneity. And actually, a lot of these loci, this one here, and so on, are actually loci for atherosclerosis, right? So we have loci that affect cholesterol levels, that causes atherosclerosis. There's myocardial infarction, and then that causes heart failure, right? So we have a lot to learn about heart failure in terms of the genetics. And that's what we're hoping to do with our studies. So the model that I'm going to be discussing today is a model that was developed at Joe Hill's lab at the University of Texas Southwestern. And it's what they call it a two-hit model. It involves metabolic stress, that is a high-fat diet, which induces obesity and insulin resistance, and what they call mechanical stress, which is a nitric oxide synthase inhibitor called LNAME. And this influences elevates blood pressure, it causes hypertrophy, aortic stiffness. And together these mimic, so what we think of as heart failure with preserved ejection fraction. There is impaired diastolic function. There's left ventricular hypertrophy, exercise intolerance, but the ejection fraction is not affected. Okay? And let me show you then, just in a black 6 mouse, as we've set it up. This is the model. When we administer this high-fat diet plus LNAME, the mice become obese, right? They become insulin resistant. Their exercise ability is reduced. They can't run as far. Left ventricular ejection fraction, however, remains the same. And here's unpaired diastolic function. That's the key trait we're going to be looking at. And it's, you can examine it with pulsed wave echo. And I suspect that you cardiologists know more about this than I do. This was done by Yang Cao, a fellow in our lab. And so what you're measuring here with E to A is sort of the velocity of blood, early to late diastole. And you can see on the HFpEF diet how much it changes, right? E to A ratio goes way up on the HFpEF diet. I don't know if this is sort of, and so you can see the E to A goes up. This is an indication of diastolic dysfunction. And here is E to E prime. It's similar, but you're measuring the velocity of the wall here, changing the wall. So both of these measures suggest diastolic dysfunction. Okay, so in our experimental design, then we take mice, various mice. We've done various kinds of study. We put them on a high-fat, high-sucrose diet. We put them on, treated them with isoproterenol. We've done a variety of studies. Including in a subset of the mice, this two-hit model. So here's what we usually do. We at about 8 weeks of age, we do echocardiography. We put them on the high-fat plus L-NAM diet. We do echocardiography at about 5 weeks. We measure weight here and so on. And then at 7 weeks, we sacrifice the mice. We do echocardiography. We look at clinical and molecular traits associated with HFpEF, and I'll show you these traits. And we do, in some of the mice, or many of the mice, we do molecular traits. In all the mice, we do transcriptomics. We take the heart, and we do RNA-seq. Right? We might do it in another tissue, such as liver or adipose. We also do metabolomics on some of the mice, and we do proteomics and so on. So today I'm going to be talking primarily about the transcriptomic analyses. Okay, so what kind of variation do we see? Well, I'm just picking 5 strains here. And this shows you roughly what we see. And, there are variations in insulin resistance and obesity. Heart weight, this is normalized heart weight varies. Lung weight varies. E to A ratio, measure of diastolic dysfunction varies. So does E to E prime ratio, right? So we see variation in the clinical traits, we see variation in the molecular traits, and now we try to put it all together. And we can use this now to study, we can identify causal genes, we can model biologic networks and so on. Today, I'm going to tell you about how we use this sort of systems genetics approach to understand how one organ talks to another. Okay, so now let's get back to a liver heart endocrine circuit. How did we identify this circuit? Okay. Oh, so why did we even look? Well, there are many studies suggesting that there's an association between fatty liver disease of all kinds, and heart failure. And this is one study. There are quite a few studies. This is just one I picked. So here we're looking at about 1,668 people that don't have not alcoholic fatty liver disease, 159 that do. And we can ask now, is there a relationship between left ventricular mass and fatty liver disease? Well, yes, a strong relationship. Left ventricular hypertrophy, this is normalized to body weight, I guess. And you can see that, yes. E to E prime ratio, this is diastolic function, yes. Ejection fraction, yes. Much less significant, but still significant, right? So, liver disease appears to contribute to heart failure. I think there's evidence also that heart failure can contribute to liver disease. But we're interested in looking at this. And so what we did, we actually did all kinds of sort of modeling. But what I'm going to tell you about is asking, are there proteins, endocrine factors secreted by liver that affect the heart and affect the heart sufficiently to contribute to heart failure? Okay, and this is the way it works. This is kind of the complicated part of the talk. I hope I can make it clear. If you have any questions, just ask. So across all 100 strains of mice, we have global gene expression in liver, and we have global gene expression in heart. And now we can ask, does expression of a particular protein in liver perturb the heart in some way, right? And what I'll show you is just looking at global gene expression. Across these strains, almost every protein varies. So let's say we have a protein here in liver, a secreted protein, it goes up and down across these strains. And we can ask, is it correlated with global gene expression in the heart? If indeed it's an endocrine factor that is acting on the heart, perturbing pathways, then you might expect there would be some sort of a correlation between the expression of that protein and expression in the heart. We can do this in various ways, but now I'm just going to ask global expression. So here what I plotted is we take each protein here in liver, there are about 15,000, and we ask, what is its p-value with each transcript in the heart? And then we add all that up and we plot it. And what we see is that there's distribution here. This is around 0 correlation. By chance, we'll have negative correlation, a little bit positive. But out here, you can see that some genes are highly correlated. And what we find is that when we look at these genes, we can identify known endocrine factors. So for example, when we look at adipose and hypothalamus, leptin is right at the top, right? We can, there are many endocrine factors that are known. And what we're asking is, among these genes out here, are there some endocrine factors, some factors produced by liver that act on the heart and perturb gene expression in the heart? Does that make, I hope that makes sense. If it doesn't, just please interrupt. Okay, so we picked several of these that are novel, that are all secreted proteins. They're secreted proteins by liver, and they appear to be doing something to heart. And the one we picked is a coagulation factor. Okay, it's produced only by liver. Its only known function is in coagulation. It's a protease in the coagulation cascade, right? And it seems to be perturbing liver, heart, right? We actually identified 2 other proteins and validated them. One of them is being worked on by a graduate student in the lab, Michael Yuda. But this is the one we decided to follow. And the reason we wanted to follow it is we're interested in heart failure with preserved ejection fraction, right? And so if across the 100 strains we look at, or no, this isn't across the 100 strains. This is just a subset of those strains. If we look at chow versus HFpEF, we can see that factor 11 is reduced under HFpEF conditions. And also what we find here is what was particularly interesting to us is that factor 11 is inversely correlated with this measure of diastolic function. The lower the plasma, the factor 11, the worse the diastolic function. So we have this relationship to diastolic function. And so we decided to try to understand how factor 11 interacts with the heart. Okay, at first we overexpressed factor 11, okay, using an adeno-associated virus. It goes into the liver, it overexpresses, and you can see in the transgenic here, in the AAV overexpressor, factor 11 is up, but it's not up like 20-fold, right? It's up about 50%. And we did that deliberately. We want to see physiology, real physiology. If you overexpress it enormously, who knows what it's doing. This is just up a little bit, and you can see the effect on diastolic function. So here's chow, the E-day ratio. You can see these 2 peaks. Now, if you put it on the HFpEF diet, you can see very, very substantial change, right? That's very easy to see. It's that ratio. If we express green fluorescent protein in the adenovirus, there's no change. But if we express, I don't know if this is in the way. If we express, yeah, let me do that. If we now overexpress factor 11, you can see it goes back almost to normal. So factor 11 has a dramatic effect on diastolic function. And here you've just seen a graph. So we look at echoes at 0, 4, and 7 weeks. And you can see, here's chow, these two bars here. Here is this HFpEF diet. Okay, it goes up. And here it is where we have factor 11. And you can see factor 11, this is GFP, this is factor 11. Factor 11 improves diastolic function, measured by E to A or E to E prime. Okay. It doesn't go back quite to normal, but it improves it. Are there any questions? No. And you can see left ventricular ejection fraction is not affected. Okay, now we also look at hypertrophy. This is heart weight divided by tail length. Here's chow diet. On the HFpEF diet, heart weight does go up, right? When we add factor 11, the hypertrophy is reduced. Lung weight also goes up, right? It gets loaded with fluid. If we express factor 11, lung weight is not increased as much. If we look at running distance, how far can the mice run? This is forced running. Here's chow. On the HFpEF diet, they can't run as far. But if we overexpress factor 11, just by 50%, right? The mice can run farther. So heart function is being significantly improved by overexpressing factor 11. Okay, here is fibrosis. We did trichrostaining of the heart. Here's chow. F11 doesn't really, it's not significant, maybe improves a little bit, but overexpressing F11 on this sensitized HFpEF background reduces fibrosis. It also reduces the expression of a variety of inflammatory proteins. We just picked 3 here, but a variety of inflammatory. So inflammation is reduced, fibrosis is reduced. Okay, so this is great. F11, if we overexpress it, helps reduce the traits associated with HFpEF. How does it do it? How could this protein do this? So what we did was we looked at the pathways. We did global gene expression in the overexpressor and non-expressor. We also had global gene expression in the panel of inbred strains of mice. And we asked, were there any pathways that were associated with F11? If we take the genes that are associated with F11 and we do pathway analysis, do we find anything? And so we found the BMP pathway appeared to be perturbed. BMPs activate these Smad transcription factors. And you can see these Smads, if we overexpress 11, are overexpressed. These are just Western blots. TNF is down a little bit. This is another Smad, it's not affected. AKT, NF-kappa B, this is just actin control. So we found that this pathway, this BMP pathway, although you'll see BMP7 is not affected, but the downstream activation of it results in activation of Smad, that is activated. Okay, so Smad 1,5 goes up a lot. We know this is controlled by the BMP pathway. TNF is just an inflammatory protein. F11 reduces it, as we've shown you before. These other pathways, candidate pathways, were not affected. So we were interested in looking at this pathway. And so BMP pathway is activated by bone morphogen proteins, particularly in this case, BMP7. And we're asking if F11 somehow activates this pathway or increases the activation by some other mechanism, right? And the way we first looked at this is just using an inhibitor of this pathway. And this then phosphorylates SMADs 1,5, and now you get, these are active in transcription. So we added an inhibitor called DMH1, a previously known inhibitor, and we asked, if we inhibit the BMP pathway, do we affect diastolic function? And here what we have is just GFP, F11 rescues. But if we add this DMH1 inhibitor, we no longer see the rescue, right? So that suggested to us, it gave us a hint that the BMP pathway might be involved in this circuit. Okay, then the question is, how does F11, this coagulation factor, activate BMP signaling? Well, if you remember, I told you factor 11 is a protease. So one thing that we thought about looking at immediately is protease activity. If we inactivate the protease of F11, is it still active? Right? Actually, there are common mutations of F11, inactivating mutations in Ashkenazi Jews. And so people have identified these mutations before. So we introduced these mutations by in vitro mutagenesis. We did both the mouse and the human factor 11, and we asked, could it still activate the SMAD pathway? So here's control. If we had normal F11, you can see SMAD 1,5 phosphorylation goes up. If we had the mutated, it's inactive. So you need active F11. Okay. So here you can see, again, diastolic function. It's improved by adding F11. If we overexpress the mutated form, it doesn't do anything. And the same thing with E to E prime ratio. Right? The normal improves. The mutated, it's not a significant difference. Right? It doesn't do anything. Okay, so it looks like it's the protease activity of F11 that is somehow activating the BMP pathway. How could it do it? Well, BMP7 is actually secreted as a precursor from cardiomyocytes. It's secreted, it binds to the extracellular matrix, right, as an unprocessed monomer. It's not active, and it binds to the matrix. And it's activated by being cleaved, right, by F11 or other pathways, cleaved, releasing the active dimer and monomer. And then now these are released from the extracellular matrix and are free to activate the BMP pathway. And you can see overexpressing BMP. Forget if this is in tissue culture or in vivo. It works in both cases. You can see that we get activation. We release the active BMP7. It wasn't increased in expression. What we're doing is activating it. So this is a figure that came with our paper. It's an editorial by Drs. Tong and Joe Hill. And it's a nicer figure than the one we made. So I'm going to show that. So what happens is the liver makes factor 11. It goes into the circulation. It acts in the coagulation pathway, but it also finds its way to the heart. And there it can cleave this BMP7 that's stuck on the matrix. And here's the matrix. Here's the cardiomyocyte. It cleaves the active dimer from this other part, releases it. The BMP7 that now interacts with the BMP receptor That phosphorylates SMAD1/5. That activates transcription of many genes. This has previously been studied. This reduces inflammation, it reduces fibrosis, and it reduces diastolic dysfunction. Okay, I hope that was clear. Okay. So just to be short, because mostly we've been overexpressing F11. What happens if we reduce its expression? So we got the knockout mice. The knockout mice are not very healthy. So we used heterozygotes. Okay, one normal copy, one mutant copy. So here's F11 RNA in the wild type. And as you expect, it's about 50% of the levels in the heterozygote. It's not expressed in any other tissue except liver. And so now if we look at SMADs, right, as compared to, this is just chow, and this is on the HFpEF diet, the wild type and the F11, you can see that it's reduced about 50% compared to the wild type black 6 mouse. Right? So it doesn't activate SMADs as well. The diastolic dysfunction measured by E to A is worse. Lung weight is increased and running distance is decreased in each of the heterozygous mice. So using a knockout mouse is exactly the same conclusions as overexpression. Overexpressing F11 improves diastolic function. Reducing F11 worsens diastolic function on this sensitized background again. Right? Okay. So everything's consistent. Okay, now we ask, does F11 affect heart function in humans? Right? And we were very fortunate to develop a collaboration with a group in China. This is a group that Yang Cao did her MD or Ph.D., not MD, Ph.D. in China. And she knew these people and they were interested in collaborating. So they had a cohort of HFpEF patients. And so we measured F11. And what we found was that F11 didn't differ between HFpEF and controls, right? But it did associate in both controls and patients with diastolic function, right? Significantly, right? And in both. If you do the combined p-value, it's very, very small. So all by itself, right? All by itself, it isn't enough to cause diastolic dysfunction, right? There are other factors, obesity, for example, is a very important factor in diastolic dysfunction. Sex is important. There are many things that contribute to diastolic, but given those characteristics, it has a big impact. And what it's doing, even in individuals that don't have heart failure, normal individuals, it affects diastolic function. So, and we're interested in looking at exactly what are the changes in controls. We've done that a little bit. It looks like it's, again, it reduces fibrosis, it reduces inflammation a little bit. So, so this is, I think, strong validation that F11, that we have the same axis in humans, and that in humans, it appears to contribute to at least diastolic function, right? At least diastolic function. Okay, what are we doing now? And one question we had is, why do we have this circuit, right? What's the purpose of having the liver talk to the heart in this way, right? And we assume, it's probably got to do with coordinating metabolism, which probably is true. But we discovered something interesting if we look at overexpression or underexpression and so on, and we do gene expression. what are the genes that are perturbed by F11, in this case, overexpression? And what we find is biologic rhythms. Really, the p-value is like 10 to the minus 10. It's the strongest effect. Overexpress F11, you perturb genes associated with biologic rhythms. So which would be the circadian clock. Right? And you can look at the individual genes, right? The circadian clock is sort of like a feedback loop with a bunch of transcription factors, right? It controls circadian rhythm. It's perturbed by things. You can throw off the clock if you stay awake, or if you don't have sleep, or depending on what you eat. So there are things that perturb the clock. And so we think that it's affecting the clock, presumably to coordinate metabolic regulation of the liver. The liver obviously is exposed to nutrients, it knows when you eat, with function in the heart. That's our hypothesis. And we look forward to looking in more detail then at how is metabolism of the heart affected by factor 11. As I showed you, factor 11 is even acting in individuals that don't have heart failure. So it's something that is involved in heart function. So with that, I'm going to end. I'm going to tell you, Yang Cao has led this study. She is a totally brilliant scientist, young scientist. She was fortunate enough from these studies to identify, to get an offer for a faculty position, which I think she will take. Other people in the lab are Yuchen Wang, who happens to be her husband. He's also extremely talented. Sicheng Zhao is our lab manager. He helped. Yonghong Men and Calvin Pan also helped in the study. And of course, there was an interaction with all of the members of our laboratory. I want to thank Human Alai, a professor and former graduate student who's now at USC for informatics, Ling Jiang and Tang Tiao Li in China for the human studies, and Marcus Selden at UC Irvine, who developed the systems genetics approach to identifying novel endocrine factors. And Marcus has applied the same method very successfully to a variety of novel endocrine interaction circuits. Anyway, so with that, I will end, and thank you very much for paying attention, and I'll be happy to answer any questions if there are some.
Thank you, Dr. Lusis. That was an amazing talk. I have a couple questions, or maybe others as well. But for the, so it seems like there's you presented two different mechanisms by which there could be, HFpEF formation with this pathway. One is through inflammation through the Smad, and then also through dysregulation of the circadian rhythms and others. Is as well as I understand that correctly?
Yeah, Christopher, I think they go together. My guess is that it's just a guess that, well, no, you're right. They probably are separate. What I was thinking was that well, maybe not. The BMP pathway perturbs these transcription factors that do many things, and I think that the things that the SMADs do can in turn affect the circadian rhythm. That would be my guess. It's possible that F11 is doing something else besides activating BMPs, the BMP pathway. But my guess would be that it affects the BMP pathway, which in turn affects SMADs, which in turn affects the circadian rhythm. But that's something we can test. That's a really good point. We can activate the SMAD pathway in other ways, and we can ask whether the same BMP or circadian factors are affected, which would be a good experiment. I like that.
That's great. And then I think another question is just a little separate, but I think it was pretty striking how diet alone can invoke a HFpEF morphology or a phenotype. Was there anything specific about those types of mice, or were those just standard mice and you're giving them a HFpEF diet?
They're just standard mice, but again, it's high fat. But a key part of this to get the HFpEF phenotype is the LNIN, nitric oxide synthase inhibitor. You know, nitric oxides cause vasodilation and so on, and they are key in inducing hypertension and sort of vessel stiffness and so on, heart stiffness. So if you just use a high-fat diet, you don't develop HFpEF.
Oh, I see. Okay.
So yeah, so we didn't develop that model. That was developed by Joe Hill and his colleagues at the UT Southwestern University of Texas. But that's key. It's not just the high fat.
Okay, so LNIN.
Yeah.
Hi, I have a question, Jake.
Oh, hi, Tintud. How are you?
Good, how are you? Thanks for the great talk. Do you think the F11 has some threshold levels? Because your wild type and F11 knockout, that's only a little bit different. Like wild type, I see like 0.6 fold.
Yeah.
Knockout is only 0.4. But the phenotype is, dysfunction is clear.
Yeah. Yeah, I think that's a good question. I mean, there's no reason it has to be kind of a linear relationship. There are people that are homozygous for null mutations of F11, and they don't have a very serious bleeding abnormality, but they bleed. And I don't know. That's a good question. That's something that it would be very interesting to study. That sort of dose response curve.
Yeah.
Yes.
And your overexpression is only 50% fold, right? Increase.
Yeah, it's a very small amount. Yeah. Yeah. So just going twofold or down to 50%, has a pretty big impact on the heart. It's very surprising, I think.
Yeah.
Yeah.
So it has a coagulation effect as well?
It does. It's a coagulation factor. It activates factor 12.
I see.
And, some deficiencies of certain coagulation factors have a really, really big impact. This one, the impact isn't as large, so which may be a clue as to another function that it has. I don't know. But it's a coagulation factor. It's only made by liver. And, until our studies, it was the only known function of F11.
I see.
Yeah.
One more question. Do you see
Sure.
any effects on vessels?
Any what?
Vessels, like aorta, artery walls.
Oh, does it affect sort of atherosclerosis or thickening?
Yes.
We haven't looked. Our lab should be doing that. We just haven't looked.
Okay.
I mean, based on where BMP7 is expressed, it looks like it's acting on cardiomyocytes.
I see.
We don't know that it's not acting on other cell types, actually.
Mhm.
It could be. One of the things, people sometimes ask, why is it affecting the heart? Why doesn't it affect all tissues? I should have mentioned this. And the heart is really, very different from the other tissues in that it has a lot of the inactive BMP precursor. If you just do a Western blot, there's a lot of it sitting on the heart.
Yes.
There's a little bit in adipose, very little in other tissues. So the heart, for some reason, expresses a lot of this precursor, BMP7 precursor, that's just sitting there. But other tissues do not. We really haven't looked at aorta. It's possible that aorta or smooth muscle cells or something could express it, and that would be worth doing. Yes.
So this F11 interacts specifically with BMP7 and not other BMPs?
Well, we've checked some other BMPs and it has no effect on some others. Actually, the factor 11 cleavage site is very similar to the site of factor 12 that F11 acts on. That was one of the clues for us that that F11, would be cleaving BMP7. But as far as we can tell, we haven't looked too extent. We've looked at a number of other BMPs, no effect. Of course, you're interested in calcification and all these things, yes.
Yeah. Because other BMPs are very prominent promoter of calcification.
Yes. Well, that would be great to look at, I think. Yeah, I think it would be worth it.
Thank you. Very nice.
Yes, thank you.
Hey, Jake, can I ask a quick question?
Of course.
It's a fantastic talk as always. So as you know, BMP7 binds to matrix proteins, right?
Yes.
Do you know if you factor 11 actually cleavage-free BMP7 or has to be can bind to matrix that can prevent BMP7 to be cleaved by factor 11?
Yeah, it's again, we didn't discover that F11 precursor binds to matrix. That's been known for 10 years or something like that. And it's also known that when F11 is cleaved, it releases the active dimer. So it looks like the other part of the precursor is what binds to the matrix. And when it's cleaved, it's released. And presumably it interacts with the BMP receptors in the heart is because it's right there. I mean, part of it probably floats off and so on, but it's right there. Was that your question?
Yes.
Yeah.
Another quick question: that if you overexpress F11 by injecting virus, do you see any F11 overexpression in the heart too, or just in the liver?
Yeah, well, we use a virus that has a liver-specific promoter.
Yes.
So I guess with the virus, you could always have just a little bit elsewhere, but it's a liver-specific promoter, and it's sort of the AAV is sort of tropic for the liver. So my guess is that there's very little expressed elsewhere.
So just my final question, and as you know, aging is a very important risk factor for HFpEF. Have you assessed the effects of aging in this mice to see the effect?
Yeah, that's one. That's one of the things that was asked by reviewers. So the Hill model is one model for HFpEF, but there's another model that's like obese aged mice. And so the reviewer asked that, and we checked that, and F11 also protects there.
All right, thanks.
So we did that. Yeah. Aging would be another interesting dimension here to look at, I think.
Yeah.
And how F11 is regulated. I showed you on a high-fat diet, F11 goes down, but we don't understand, really anything about the regulation of F11. So there are a lot of interesting things to do.
All right. Thanks, Jake.
Thank you.
Oh, hello, Jake, and
Hey, Suze, hi.
how are you?
You already heard this once before.
Right. And I got even more impressed the first time because I had the opportunity of looking at your paper.
Oh.
And seeing some of the data. And for the fellows, I just would like to say, this is an amazing opportunity of seeing this intersection in between the basic science and animal work and clinical science, and also to believe that echocardiographic assessment of diastolic function is not just valid and important in humans, but even in mice. So I was looking at your model of diastolic dysfunction, and it is one type of there are different degrees of and types of diastolic dysfunction, and I think that this resembles one that is like due to abnormal or restrictive physiology. So it's a pretty advanced degree of diastolic dysfunction. Not the most typical that we see. We actually see in the clinical, in our patients, and other types that are a little bit milder, like abnormal relaxation, which is the first degree or mild degree of diastolic dysfunction, or pseudonormalization abnormality, which is the second degree. But my question comes into when you're trying to establish this parallel in between the mouse model of this restrictive physiology and the human, I wonder if one of the reasons why you're not seeing the association coming out significant in the humans is because you're using a parameter that is you know, this E2E or this E2A ratio.
Yes.
For example, it's just detecting one type of diastolic dysfunction. Right?
Oh,
You
yeah.
could have a completely normal E2A ratio or even an inverse E2A ratio, and then you will have to rely on others. I believe that you also use a E2E prime.
Yes.
Another good measure. But could it be that this factor 11 is actually important in some types of diastolic dysfunction or some subset? And when you're looking into the bigger population, so the subset gets diluted. So,
Yeah.
so really, so it's sort of like a, you know, it's being like a, I don't know if diluted, it could be, or mask, you know, by the other part.
Yes.
You know, in clinical science, actually, we see it. We'll see like this moderate degree of diastolic dysfunction. It actually looks completely normal. And then if you go to and try to do associations, you will not see anything. So if you you didn't see that problem in mice, because the mouse gave you just a model of just one type of diastolic dysfunction. I mean, this is restrictive physiology.
You know, that's a very interesting point. I think, you know, again, I wish I was a doctor and I actually had seen this in living people and stuff. I think what I had assumed was that it's one factor of many. You know, you have people that, I mean, if you look in the population, just like people that have atherosclerosis and that don't, you know, cholesterol isn't all that different, right? But we know the cholesterol is incredibly important and so on, you know, and so I think it's just one factor of many, maybe several at least, important factors that contribute. But I think that that's a very good point. HFpEF is obviously very heterogeneous, and you know, I was on an NIH sort of. There's a certain aspect of NIH that's trying to get sort of industry collaborations on HFpEF, trying to get industry involved in studying it and stuff. And it was really clear. I mean, there are some, you know, all kinds of things that can contribute to a HFpEF type phenotype. And so that certainly would reduce the power to see it in just a straight comparison. Yeah, thank you. I'd be very interested. I should have talked to you about this earlier. You know, exploring those other aspects that you know might help us understand this further. Thank you.
Yeah, beautiful. Thank you.
Thank you.
I think if there's no further questions, thank you very much, Dr. Lusis, for
well, thank you very much,
amazing
Christopher.
talk.
Yeah, thank you very much.
Thanks.
Thanks, Jake.
Thank you.
Thanks,
Thank you
Jake.
so much.
Yes.
Thank you.